I think this is going to be like, so the objective behind developing the ONT barcoder software was to reach out to everybody and make it easy for running analysis. So I don't think it's going to be that complicated to run. So what I'll do is mostly talk through the slides and and explain the various variables that can be changed and what is behind the scenes happening in each of those steps. Yeah, I'm from the Center for Integrative Biodiversity Discovery in Berlin, and we are a relatively new center who are doing a lot of large-scale species discovery projects. So we are like basically doing large-scale barcoding and going on to integrative taxonomy efforts. So I thought, firstly, since yesterday a lot of the molecular steps were done with ONT, I thought we can first look at the ONT's video of what Nanopore's internally doing with the sequencer. So essentially, when the library is loaded, you are having a DNA molecule going onto the flow cell, and then during the library preparation, you are attaching a protein molecule that tethers onto the flow cell. Yeah. And the main thing that's happening here is that your double-stranded DNA is turning into a single-stranded molecule. And what then happens is the changes in electricity are being read. And this is the raw data that was being generated yesterday from the flow cells. So this is the raw signals. And the intention is very much to go from these raw signals to your barcodes. And for that, what ONT provides is like a bunch of tools, and we don't deal with the conversion of the raw signals to the data set. That's the basecalling process. And what has been happening with ONT is it has been changing and evolving quite rapidly. So I think what I would say is that there are three main things that have been changing in ONT. One is like the flow cell. So yesterday, the sequencing was done in R9.4 flow cell. This was actually introduced back sometime in 2016. And subsequently, in around 2020, they introduced the R10.3 flow chemistry. And then you have, in 2022, R10.4. And the main thing that has been changing in ONT chemistry is the type of data set. So like you saw the pore there, and then you have a DNA molecule going in. And the changes are being read, and the biggest challenge had been that it would create some sort of systematic errors. So you are having like a homopolymeric stretch. You're not able to actually see the changes in the electricity because you don't know how long that has been happening. So these would often create these types of systematic errors. And this was a challenge with CO1 barcoding because CO1 is does have homopolymers. So if people have examined these data sets, they'll always see that there are these regions of poly-As and poly-Ts. And this caused some of the initial challenges that we had when we were trying to develop this pipeline. But then when they developed the R10.3, what they came up with is a dual header. So you can actually measure not just the change, but the molecule is going between two headers, and that tells you the distance between the two, and that can help you basically give much more random set of errors. So basically the error rate did not change too much between chemistries of these two chemistries, but the nature of the errors changed. So you did not have as much systematic errors, and this allowed really the barcoding to improve. The other thing that has been changing quite a bit, I mean, one note is that the exact details of this change are not very clear. I've been trying to figure it out, not sure, but one of the things that they do mention is that the enzymatic aspects of the chemistry has changed in the R10.4. But this has been the most dramatic improvement we have seen. The second type of improvements have been happening in terms of base calling. So they have been releasing different models, and models are basically trained with like references. So you take these electrical signals, you're mapping them onto, let's say the human genome, which is well studied, and then you basically improve the quality of determining the new data sets. So again, from fast base calling, which was introduced back in 2016 or so, they moved to high accuracy base calling, and lately they've been doing super accuracy. Yesterday we tried to set up with the super accuracy initially. We still have the super accuracy base call data, but the initial setup didn't work, and I'll also take you through why, because I think these are the kind of challenges that's commonly happening when actually people are trying to implement ONT barcoding on their own. So with fast base calling, the good thing is their device MK1C can handle it, and it can do the base calling as the sequencing is going on. And you can set it up in Minnow itself. So you just have to enable live base calling. Minnow will generate the fast base calls. With high accuracy base calling, this particular device can do it, but it can take a week to do it for a full min ion flow cell. Flongle is much faster, so you can probably get it within a day, but with the full run of like something like 10-15 million sequences, this is not the device you would like to use for the practical purposes. So getting something like a good GPU laptop or a computer would be good. And there are recommendations, and there's a really good one person, I think Benton, he has like really explained this, which GPU you want to buy, and I think that's available publicly if we ever want to see that. With super accuracy, this device just couldn't handle it, so you just have to rely on something external, which is what thankfully Natasha provided, and so we could do yesterday these base calls. Not only did base calling change, they also changed the base calling software. So again, back in 2017 or so, they had Albacore. Albacore was a CPU-based base caller, and it could only do something like the fast base calling. And then with Guppy, you had the GPU usage, and now, very recently, everything seems to be moving to Dorado, which can nearly double the speed. It also uses files which are more written more quickly, as well as they're much smaller in size. So one of the challenges with ONT data is that it's huge. Yes, basically like a min ion run a full run can take up something like 200 to 300 GB per run. So you're basically, if you're like a routinely sequencing this, something like once a week, you get like terabytes within few months of usage. So I think space reduction has been a major challenge when it comes to data handling, and I think they're moving towards that. So let me just so yesterday I used Guppy because I think it would have taken time to convert files for Dorado, and I wasn't very sure whether the latest upgrades would work. But Guppy is still quite standard. And during the Guppy, when you are setting it up in Minnow itself, you can set up the base calling like you saw yesterday. The main challenge we had was that there is a CUDA driver that needs yet to be upgraded. So if you ever face like, if you've bought the right computer and things are not working, usually it's a driver related upgrade that you need to do. And yeah, they have a toolkit and that should take care of it. So we just downloaded that, installed it, and it worked fine. So coming back to the older flow cells, so this was how the error profile for an older flow cell looks like. So this is mapped onto a 418 base pair fragment of CO1. So 40 is essentially 10% errors. And what we were finding was when we were mapped a data that was coming in 2019, and these are raw reads mapped onto reference barcodes. This data set is like about 3,600 barcodes, and what you could see is that it's about 10% error, and the dark blue here is like if you allowed for one base pair error in tag, and light blue is the two base pair error in tag. So it was not like it was a demultiplexing issue that was causing this. It's clearly like a error profile. But back then we started developing this tool called mini barcoder. And this one is basically was a command line tool, and this is when we mostly resolved the demultiplexing challenges that ONT data presented. And we compared the resulting MinION barcodes to about, for a data set of about 7,000 barcodes, and it was at about 99.9% accuracy here. So the main thing was we had to implement a lot of correction steps. So to go from something like this to this, it required a lot of polishers, a lot of alignments, and then final quality controls. So at this point, we estimated we could do something like 500 to 3,000 MinION barcodes per flow cell with a cost of 50 cents. And this, of course, is mostly driven by also the chemical costs. So you are, this includes the price of the PCR, the plastics, and such. So yeah, but we were able to do the decent characterization. So from a Malay strap, which we saw assembled, this was, we basically barcoded something like the 7,000 flies of this particular family, which is a hyperdiverse Diptera family. And from merely 8 weeks of sampling, we could find nearly 650 OTUs, which was quite a large diversity if you think about it. So, and these OTUs were virtually identical to the Illumina OTUs, so we saw no real difference in the interpretation. But the challenge at this stage was really the bioinformatics. It was the classical bioinformatics pipeline. We had like lots of scripts, lots of dependencies, and installation was often a challenge. So the other problem was to account for systematic errors. We used the property that CO1 is a translatable molecule, and because of that, we could kind of gauge where the frame shifts were occurring, and we introduced ambiguities because of that. So this pipeline had this problem that it was including lots of ends, which is not desirable for your barcodes. And because we had a lot of software dependencies, one of the ones that we used was this Recon, which was a polisher for nanopore data. And that one, and basically we used like multiple consensus to correct and eventually got to a decent answer. But we wanted this particular pipeline to be a bit usable for everybody, but at that in 2018 this was not really doable. So in 2021 we started working on the 10.3 data sets, and this is the error profile you could see. So from 40 we are somewhere peaking around 20, and that one is having about 6% error rates, this and the nature of the errors not being systematic, suddenly we needed like much fewer error correction steps. So we could just like work with very really like simple alignment tools. And this is when we started decided that okay, we can start building the ONT barcoder GUI, because you don't have to have installations that would be difficult. And you could do like variable scales of barcoding. So this is the coverage assessment. So basically how much data you need for how many barcodes. So this is like the two runs here, this and this are the Flongle runs. And this generated somewhere about 200,000 to 300,000 reads. And we could get something like 250 or 200 barcodes with Flongles. And we could go up to nearly sequencing 8,500 for a full MinION run. We subsequently, internally in the lab, we were able to hit to 13,000 or so. So that's where we would usually sequence by the time that many products were available. So this is like the general estimates we had at this stage, which was the 10K per MinION flow cell and 250 per Flongle. In a way, it was also a management thing, because like Ben was mentioning about tagging yesterday. So when you order like a 96 well plate of tags, it's like 96 by 96, and we could get like 9,900-ish products multiplex. You could, of course, do more plates and more barcodes, but I think we never had that need, really. At this stage, we could hit about 99.99% accuracies. We resequenced some of the older data sets that we had done previously using the 10.3, and that gave us also references. And these references were generally obtained either with Sanger or Illumina. So it's not within in Minion comparisons. So this is the quality we are seeing for 10.4. So you are now virtually within 4 base pairs. Most of the data is there, which is less than 1% error for the 400 base pair molecule. And this is why I was saying that most of the stuff that we actually ended up doing for correcting was not really necessary because the technology evolved that fast. And you can also see distinct improvements in demultiplexing rates. So we are hitting routinely about 50% demultiplexing rates these days, which is, I think, also interestingly what I observed in today's data set. So it'll be nice to see. I think partly this is also driven by the models of basecalling that is getting available. So maybe we can start playing with the data now with the ONT barcoder, and we can open the software. And now the recent version, we have 2 modes, which is like having conventional barcoding, which is like the standard pipeline that we would be using. There's also the real-time barcoding, which I will get to towards the end. So if the data set is not, because it's just internal. So I have the link though, or if it's in the thumb drive. Yeah, of course.
So we have always wondered why the demultiplexing is only around 50%. Could you explain a little bit more about that?
Yes, of course. So part of the reason is, of course, the errors. Like I think, so for us, we want to get as much accurate data into the bins as possible rather than getting more. So you can, of course, maximize demultiplexing rates by including more errors. We always demultiplex both pass and fail. So it's not a QC controlled file here. So you can probably get more if you did like a quality filtering in your data set. So if you did like a Q cutoff of 15 or 20, you do get more. But we generally find that getting rarer specimens is more important for us than to get more reads is more important for us than getting more QC data. We tried calling the barcodes with and without that filter, didn't make a huge difference in the final barcodes accuracy. So yeah, overall, it's the length. The short length does compromise your demultiplexing rate also. So you are ultimately, if you had like a really long index and you could allow for a lot more errors, you can probably demultiplex accurately, but then your PCR is more complicated basically. So yeah.
And you use the failed reads as well? Because we only ever use the pass reads.
We usually use the fail reads. Yeah, I mean, there's a lot of literature as well on this that Q scores in Minion were not really mattering. Demultiplexing rate does differ in that sense. So there is some difference overall in the accuracy of the read, but the positionally in the SNPs, when you're looking at it, it was not like every the Q score was positionally impacting the consensus. Yeah. Okay, so have everyone managed to open? Yeah, so the process is rather simple. You can just select and start. And there are 3 modes in the software where you have like, the mode 1 is the one which works when you have like starting from raw FastQ to demultiplexing. You can also feed in demultiplexed files in the second mode. So in case you want to use another demultiplexer, you just have some formatting requirements that needs to be followed. This is for some output modification, error corrections if you just want to perform some error corrections finally. But so we'll just use the mode 1 right now, and you can basically drag in your all indices file, which is your demultiplexing file. And you can open that file and see what it's like. I will also explain that in a bit, but you could also start the run and we can go through the files together. So once you have the files there. You can, it will basically open up a dialog to show the various options. I can go through each of the options in the slides, but you can also feel free to start the run or wait till the slides are over. The analysis is quite fast for this data set because it's quite small. So yeah, so basically what you need is like these two main inputs. You have like the FASTQ file and the demultiplexing file. The default parameters will work for this run because it's a 658 base pair molecule and the defaults are set at the CO1 standards. But you may have to modify some of these if you are doing something different. So in terms of what the software will be doing, so yesterday Ben explained the indexing strategy. I'll just revisit to show the structure of the molecule. So here you have like the tags that we have designed, and then you have the forward and reverse primers. And then what you order to the IDT or something will be basically like this. You have a tag extended to the attached to the primer here. And what we want to do, of course, is always keep that information separate because we are pretty luxurious when it comes to finding the primers in the molecule errors wise, but we are pretty strict over here because we want a quite precise binning of the specimen. So this is what that all indices file, if you open it, it will look like. So what you have over here is the first field is your specimen code. You have the forward tag with the forward primer, reverse tag with the reverse primer over here. So just keep that information separate in a CSV file, and that's all that's needed in terms of your specimen information. So analysis wise, we have two main stages, which is demultiplexing, and then you have the consensus calling. In terms of demultiplexing, what we are doing is basically looking for the primer first in a read class, and then classifying the 5' end of the primer to the specimen specific bin. And so basically, okay, I think this has been started. Okay, so basically this is the configuration you must have seen before. And some of these parameters are relevant only to the demultiplexing stage here. So this is basically your minimum length parameter and the expected length parameter. This is your barcode length. And this is basically something that we just usually recommend that you can go with the barcode length. You can also really, if you have like lots of multiple bands in your PCRs, you can try to narrow the window and filter much more strictly if you want. So I'll just show you an image of like how raw reads look like. So this is like one of the worst data sets we had. So I thought we should start with like why these parameters exist. So you can see why some of the demultiplexing rates also fail because you have a variable length. I think some of it has to do with existing primers not being removed. Some of it also has to be terminated sequencing. So it's not that every molecule is sequenced to the end. So you get something like this. So if you are having a 658 base pair cutoff for a 730 base pair molecule, which is like your barcode plus primer plus tags, this is the length profile that you would be selecting for by the minimum length criterion. And so you're basically discarding all the shorter stuff. The next thing is something that we observed routinely happens in library preparations when we are preparing MinION libraries, which is that if your cleanup after end prep is not good or your termination hasn't been good, products can self-ligate. So which is why you see these really clear profiles of two barcodes being ligated to each other. And this is why we have the expected length and the windows to split the reads. And this is basically looking at which region for the we can aim at to like sort of start assessing the second piece as well. so that we can split the molecule, start looking at the overlapping region, and search for primers there. So this is like what we think might be like ligated products. And the next two parameters here are like the window for primer and tag search and the tag mismatch. This is quite intuitive. So if we are looking at primers, they are going to be something like this in a particular fragment. And this is in reality how the data looks like. This is a real raw read, and a primer is aligned to it. And what we do is basically align with quite liberal criteria. So and then I think it's about an error of 10 bases that we allow in a molecule of 25. And then we fish out this particular region. This is mostly done because we are not aligning each tag individually, largely because of the nature of our barcodes, which are very short. So we want to be extremely precise. And once we fish that out, we match it to your index information. The index information is also stored along with its errors, so like the mutant tags basically, and then matched to those mutated tags. And so yeah, that's basically the process of demultiplexing. So if you have started the run, hopefully this would have gone through the demultiplexing by now. I'm not sure, but is it running still? It's demultiplexing right now? Okay, cool. So the software, when it's running, it will actually start printing out what it's doing. So it's actually just describing these splitting and the numbers that you're getting. So you're having these raw reads, how many are like in the correct range, how many are split, etc. Yeah, that's right. I think it's the Mac. The Mac does perform a little faster, yeah.
Maybe it's just a Windows thing, but I find that it always pauses for a while at 72,004. Is there something in the software that says when it gets there, take a break, and then come back again?
I think it is recalibrating the processors at that time. So it is parallelized, so it's like trying to find, and Windows has that issue. It's not so, some of the older computers especially are not so great at handling if you have SSD, it tends to work better, yeah. Yeah. So what you can see here is your statistics. So you have like these number of reads. This is the demultiplexing rate. And how many bins have one read, how many have 5. And usually we process everything which has over 5 reads. And later on give you the coverage information for you to decide whether you want to accept them or not. So the next step is really the consensus calling. And here so when we tried to build the Mini barcoder pipeline, we were really wondering about the types of consensus calling softwares available for MinION, and this was mostly the genomic tools that are available. So you have like these polishers for genome assembly projects and such, and they were not giving us great data because they would usually trim away chunks of your barcodes, and we want the full length amplicon to be handled properly. So what we realized worked nearly as well as, I think as well as the consensus calling tools was reducing the gap opening penalty in MAFT and just doing an MSA and calling a consensus. But for ONT barcoder, we actually did something little, when we moved from mini barcoder to ONT barcoder, the change was basically we trained the MAFTs alignment parameters based on ONT data and use that model to align the data. So that's slightly, that gave slight improvements over just reduction of opening penalty. But so I think this is like a video because I think I want to just pause and start, which would be hard in the real live version. But after demultiplexing, you're basically getting specimen-specific bins of reads. And what ONT barcoder allows you to do, and if you go back to the menu that is there, sorry, 1 second, let me just open this again. So here you can see that when we established the coverage to be used, we did like a series of values here, which was like a iterative coverage mode. And one of the reasons for this was that when we noticed that firstly excess coverage in ONT was actually deteriorating the barcode quality, and that's because the large number of reads tend to create more noise, and we were not able to actually, when we compared the results between something like 200x and 1000x, we would never really see an improvement. If anything, we would see a decline. So we would always something like a 50 to 500x was really good quality barcodes, but this was at the stage of 9.4 with 10 and 10.3, 10.4. This is much less. I think even at 5x you are getting decent barcodes with 10.4. So the main purpose for the iterative coverage mode is basically to improve speed, because when you're analyzing data sets of 10,000, you're basically aligning 10,000 times. And so we are basically subsetting data at 25x. We are doing some QCs, and the QCs are basically whether your CO1 is completely translatable, it has very few ambiguities, and it's the expected length. So this is a criterion which can give, of course, ignores the real indels in amino acid indels, but I think this is almost like a needless step now with the 10.4, because the reads are getting quite accurate. So basically, with the QCs, we are able to judge whether your barcode is okay, and then move on to, okay, maybe this bin needs more reads. So this is essentially what's happening with the iterative coverage analysis when if you have set it up under the defaults. So it's basically going to call it at 25x with the masked alignments. Then it will go on to do something like 50x, going ahead to 100, 200, 500. After this, what we started noticing was sometimes you have like low coverage contaminants present in your data sets, co-amplifying bacteria, co-amplifying humans, and we wanted to make sure that those are not influencing, creating more noise. So we improved the initial consensus based on some similarities. So basically you have an initial consensus based on length, you map your reads back onto them, you improve the basis based on similarity to the first consensus. And this is basically what leads to the consensus by similarity settings. And one second, sorry, I'm like going back and forth with the video. Yeah. And here we just do one iteration. We don't think that this helps that much. It does improve a little bit. And I think if you notice, you would have, from my understanding, this run you had something like 80 barcodes that were coming from consensus by length. Something like, you can observe how many did you get for similarity, if anybody got that. Has anybody managed to reach there?
So the first alignment is 82.
Yeah, okay.
The number of good barcodes obtained after aligning similar reads is 11.
Yeah, so, and the final stage is really to like look at whether there are indels being created in these alignments that you're getting from these barcodes, and fixing those indels based on an MSA. This is something you can turn off. This is something that's the last step, and it's becoming quite unnecessary in the recent runs. So with the 10.4, when we looked at the consensus by length, 97.2% of the barcodes were coming at this step itself. Flongle gives slightly worse data than MinION. So this is MinION's numbers. And then you have, but you can see the issue in the earlier days of fast base calling in R9.4. We were hardly getting anything with just the length criterion. So yeah, these are the variables you might want to modulate if you want to change the speed up or get more the reads for your analysis. And this one I won't really go into, but please you're welcome to ask me because it's just got to do with how the consensus is being called based on your SNPs at a site. This is a the final parameter. This basically post primer finding that there is any you want to if the primers are found too deep inside, you can get rid of things that are within 50 base pairs of the 658 molecule. Yes.
Is there different default settings between Mac and Windows?
Is there?
Different default settings between Mac and Windows? Because I have a different.
What is it?
I just have a different number of barcodes obtained after reads.
How much is it?
9.
Ah, okay. So I think that can be a bit random because the read selections may not be identical because the subsetting may not be identical. So there is that little minor randomness between runs. We see something like, it will vary, something like 3 or 4 barcodes sometimes. Yeah, yeah. So the results, if you have reached that stage, and we can wait here if you want, comes in the results of barcoding tab once the run is complete. So if anyone's is not done, just we can pause here. Yeah.
Very different on Linux?
Linux is should be fine. I don't think it's an OS dependent thing. It's really I had to basically use a couple of different things, but none of them affect they are basically the same version of Python and such, so I don't think that would be in any way. Okay, I'll come and see. Sorry, if you don't mind.
So basically I have to
Yeah.
make it run with another line.
But I think it's fine, right?
Yeah.
Yeah.
It is work, but
The results are the same, 11, 82. So my data set is completely different. Because I couldn't make my slides in 10 minutes. Okay. So the data was compiled this morning. So the slides are from a completely different run. So you guys are working on different data sets from the one that's on the video. Yeah. Yeah, I can go back to my analysis once. Yeah, I can rerun this and we can show that. I think. Yeah. So yeah, just ignore the numbers here. Please interpret your own numbers there. But these are the statistics that you ultimately will get, which basically summarize your run. I think I can get to the files. I think, am I looking at the right files? So is this more like the run that we have, which is like the number of reads? Yeah, so what's happening in this is basically 111 filtered barcodes, at least in the Mac that I ran. Yeah. Yeah.
Again, to the point that you made at the end. You mentioned something that there can be difference of 2 or 3 barcodes depending on the read.
Yeah, so the read subsetting is a random process, right? I mean, so let's say if you have a very similar set of reads, it's going to take the first 50 that are most similar in length to the, not first 50, like random 50 that are more similar in length. And that random process can create slight differences in the number of reads.
If I run the same data set 2, 3, 4, 5 times, I may get slightly
Yeah, slightly different, possibly.
What would be the either worst case scenario? 2 or 3 barcodes?
Yeah. Yeah, we have never seen a big difference, no. Yeah. Yeah, and these are, of course, like absolute criteria applied to slightly random processes, right? So every time this is the challenge where you're having a criterion like, let's say, 1% n, and that's 6 bases versus 7 bases of ambiguity codes introduced. And that you can accept your 7 bases if you really want, but that's the outcome. It's far less variable, I think, today, but this is a 9.4 run, so that's probably also reducing. Yeah. I think the read sets you can also get from the various, all the reads that were used are stored, so you can actually trace back which reads were in there, but we can create a unique number for that set of reads that are there also. So okay, so in terms of the output, if you have have gotten to the stage of output, you can see the results. This is the results here. So this is mostly the files that you will be getting in your output folder. And the key things that you want to care about are the main barcode results folder, which has your barcodes, and the file that we recommend for is this one, which is the filtered barcodes file. This allows a bit of leeway, which basically it allows for 1% ambiguities. There's like a little bit of ends allowed in your barcodes, not too much. All barcodes basically has everything that also has lots of ends and lots of incorrect barcodes. QC compliant is completely strict, which is basically no ends, perfectly translatable, and nothing fixed. So basically this is if you are really, really strict, and there are 3 levels to this. All right, so this is the result, faster. And ambiguities, okay, so the number is the length. This is your coverage used. So if you try to call it at 25x, it would basically have many of them would have been called at the first step itself. That's why everything is kind of standardized at 25, so many of them. Some of them had 37 reads only, so at the 50x stage, it would have called it with the 37 reads that it had. It also goes into fewer reads than 25. So just to keep in mind, 25 is the maximum coverage at the subsetting stage, not the minimum coverage. So you have 17 reads, something that's been called. So that's your barcode sets. Then what you have is, of course, the output table, which is basically the copy of, if you manage to close your file, the result summary tab is printed here. Sorry, close the software rather. And the run summary is one thing that you might want to use if you don't want to just go by default recommendations and you want to play around with coverage. So it's an Excel sheet that gives you much more detailed information. So in case of this particular run, so what Ben gave me was like lots of indices. Basically, there are 500 indices. These were not used in this particular experiment, so that's why you have so many empty cells here. But not default, but it's actually efficient. Just that it's not that we didn't get reads for that. It was not there. So yeah, the various steps are here. So you have like demultiplexing, and then the barcodes that were called at the consensus by length stage. Then you have the consensus by similarity stage here. And the most probably what you want to use is the final compilation of all of that. So we basically have, you can sort your data in your Excel sheet based on coverage, ambiguities, whatever you want, and export that file. I think IBOL standard is 1% ends, from my understanding. That's part of the reason.
Yeah, so what's that, 6 or less, I guess.
Yeah. Okay, so that was the run summary. And then if you want to, so sometimes we do get like human contamination or something. So demultiplex reads are available, so you can just, you can explore those data sets. Some people have used this for meta barcoding in this way, so you can use the demultiplex data if you want for complex samples. And then of course, because we also want to retain what were the reads that were kept. you have the sort of compressed outputs of the alignments. And I can show you what the alignments internally looks like. So this is like one of our data sets. And what you can see is this is an R9.4 run. And you can see there are a lot of differences. But one of the things you will notice in our demultiplexing file is a recoding of ATGCs to EPQ. Okay, sorry, this would be reversed, but EFPQ. And this is because the MAF model that we are using accounts for strand-specific error rate. So the orientation in which the sequence is read is recoded internally. And you can just convert them back if you want to ever use the FASTQ file, FASTA files. And but then this is, I think, getting quite unnecessary. So we might want to like revisit this part a bit, because our 10 point, sorry, this should be 10.4. This is a mistake, but yeah, it's pretty much identical reads here. So with 10.4, we are getting, I don't think the messiness is really going away here. So this is a common question, because a lot of the times people are like, what is this new nucleotides that are coming up? But it's just a recoding. So I think this is about it for the standard barcoding pipeline that we have. You can play with the coverage parameters if you want. I think usually we either do a 50x standard coverage or the iterative mode we vary between these two, depending on how large our disk scale of the problem is. But there was one other thing when we started working with the R10.4, once we started looking at this particularly high accuracy data, I think today we can't analyze this real time, but we started implementing real time barcoding, because we were like, okay, if most of the reads are accurate as the data is generated, we should be able to get the barcodes. So we don't really need to wait till the run is completed. And one of the reasons, primary motivations for this was that coverages are uneven and we don't know how much to sequence. So this is the recent version of ONT barcoder 2, which can do real-time barcoding. So you are able to actually assess the barcodes as the data is being generated. So yesterday we couldn't run this because it works primarily with R10.4. So I think our data would have looked quite, you need a lot more corrections with our 9.4 data. So yeah, so this is what we have been working on recently. And so here the good thing is you can get like basically in 3 hours time you're getting over 1,000 barcodes, and you can actually stop the run here, recover your full flow cells. It'll save you money basically, because you can you don't need to. One thing to know is that the final conventional analysis still gives you more barcodes. So this is like a good estimate, but it's and it can tell you when to stop a run. But if you reanalyze the data with the conventional one, which has more iterative steps, you'll recover a bit more. So the overall, what we are able to get down to is usually 10 cents per barcode here, and the barcodes we are getting with the standard pipeline is 99.99 with and with the real time also with super accuracy we are getting this 99.98 here. This is with Flongle though, and what you see is basically again where you can kind of see that within 3 hours you could have stopped the run and you needn't have sequenced more. And species accumulation wise, again I've just mapped on the regular pipeline here. So the conventional barcoding will give you more species. So just to note, don't rely completely on the real-time barcoding data if you want a very exhaustive analysis, but if you want a quick one, that's quite useful. The good thing is you can also do it with the fast base calling, so you don't need to like get like super accuracy base call data. We're getting pretty decent barcodes even with fast base calling with the new flow cells. So I think today when you guys selected the run, if you were selecting the conventional, none of this would have appeared. But if you select the real time, it will allow you to select which sequencing device you're going for, what's your experimental setup like. So I think with MinION, your experimental setups can vary quite a bit. So you might have a GPU computer separate. You might want to base call differently in a different computer. You might want to sequence in a different computer. You might want to see, like if you're sequencing in a device like Natasha's yesterday, then you can just base call at the same time or as you are sequencing in the same device. So you can operate this software anywhere really. So you can do it in the same laptops, different laptop, and connect it over Ethernet. Yeah.
What about if you're using a gridiron?
I would need to find out. We don't have one, so yeah, we'll have to figure out. We are planning to go to P2 though, so maybe we'll have a better idea. Yeah, P2's, yeah, we are planning to get one, yeah.
That's a lot of barcodes.
Yeah, yeah. So I think with 10.4 this is doable. We have looked at data, and what we have been exploring right now is like whether dominant sequence is the right one. Simple as that, because really we have so many identical sequences. It's of course not going to be, it will have a few barcodes with indel errors. So we need to explore this more. So does that matter? Probably not for 97% clustering type of OTUs, but maybe it might matter if you're really kind of wondering about one base pair differences. So we need to figure out what works there. No. So we have worked with people who have used it for RBCL. That's been okay. Yeah, so you can use, you can change the organism setting, and you just, as long as it's protein encoding, at least the QCs, we know how to assess it. I think it's by and large quite accurate with even with the, it's just that you might have a few indel errors there, yeah. Issue for us was the homopolymer errors, and those were systematic in earlier days. So that was one of the reasons we limited it to protein coding, because we had a way to assess what's our length. But of course you can do it. People have been doing it for bacterial CO16S for a while now, the nanopore, so it's not like it's not possible, but there is an error rate there and everything. So when you're doing these massive multiplex data set with uneven coverages, those error rates vary with coverages as well. So there are it's a bit like, I don't think it's a MAFT or a bioinformatic issue, it's a data issue. And but I think we haven't explored enough with the 10.4 yet to know whether we can get to.
The local mutation rate that you get prevents you to saying, yeah, use it in IPS1 or use it in 16S.
I don't think it's the mutation rate. I think it's more the simply that our understanding is right now based on CO1. And I think mutation rate is, so it's possible that, I think anything with too many homopolymers you have to be careful of. 16S, depending on the group, has that problem. But I think with 10.4 we are seeing a fewer errors of those nature. So I can't give you an answer today for that. Yeah.
Just to be clear, when people are using things like 16S, they're using the demultiplex data set, correct?
Yes.
Yeah.
Yes.
Okay.
Yes. Yeah. So in this case, basically, if you have an Ethernet cable with the sequencing device, if you want to do it in a different computer, this can work as well. So you just connect it to your base calling device. And we have linked it to the Guppy and Dorado within your computer, so that you can, if you want to do a base calling in a separate computer and monitor this, it's also doable. So the most recent update was with the Dorado. So I think, sorry, my computer might die, but yeah, so you can combine powerful GPU in a separate computer if you are sequencing via something like this, if you want, if you have that. And that's about it. And this is like, thanks to everybody who's been.